Welcome to Ultimate Sleep Doctor. Tonight, we'll journey to a place of quiet wonder. Deep within the emerald heart of a leaf lies a secret, a process called photosynthesis. It's how plants create their food, a miraculous dance of light and energy. But recent discoveries suggest something far more complex than we once believed. Scientists are now exploring the quantum level of this process, examining how leaves might be utilizing the very fabric of reality. It's a reminder that even the simplest things hold profound beauty and intricate workings. Consider the steady rhythm of nature, a constant invitation to find peace. If you find these explorations soothing, please consider liking this video and subscribing to Ultimate Sleep Doctor. Together, we can soothe more wandering minds, one peaceful night at a time. Just breathe, let your thoughts drift. Let's begin. The story of photosynthesis, the way plants make their food, isn't a sudden discovery. It's a tale woven over centuries, built on careful observations and brilliant, though sometimes puzzling, early ideas. It began, surprisingly, with a curious gentleman named Joseph Priestley. In the late 1700s, Priestley was a curious man, a clergyman and a scientist, living in Warwickshire, England. He was fascinated by plants and their apparent ability to bring a refreshing quality to the air. He noticed something quite peculiar. When he placed a candle in a closed jar, the flame gradually dwindled and eventually went out. This wasn't unusual, of course. A flame needs oxygen to burn. However, Priestley went further. He conducted a series of experiments, placing potted plants, willow, elm, and geranium, inside sealed glass cases. He observed that the air within these cases became noticeably clearer, fresher than the air outside. It wasn't simply a gradual fading of foul air. The plants seemed to be creating something. He meticulously recorded his findings, and he was convinced that plants were responsible for removing noxious gases like carbon dioxide, which he hadn't yet identified as a specific element, from the air. He called this animal air because he reasoned that animals needed this clean air to breathe. He believed that plants consumed these dephlogisticated air, a term from the prevailing scientific theories of the time, and released oxygen as a byproduct. It was a revolutionary idea, though he didn't quite grasp the full implications. His experiments were carefully controlled, and he diligently documented the changes in air quality within the jars. He even attempted to demonstrate that animals died when placed in these oxygen-rich environments, though his results weren't always conclusive. Priestley's work, published in 1774, was initially met with some excitement, particularly amongst members of the Royal Society. However, many scientists remained skeptical, and his theories faced considerable opposition. The debate surrounding Priestley's findings continued for years, fueled partly by the scientific climate of the time. The concept of phlogiston, a hypothetical substance released during combustion, was still widely accepted, and the understanding of gases was surprisingly limited. It was a confusing period, with competing theories vying for acceptance. While Priestley had made a groundbreaking observation, he lacked the tools and knowledge to truly understand the underlying process. His experiments focused on the effect of plants on air quality, but not the cause. He was charting the consequences of a phenomenon he couldn't quite explain. Fast forward a few decades, and we encounter William Blackman, another scientist whose work built upon and challenged Priestley's observations. Blackman, a self-taught botanist, developed a particularly intriguing approach. He was interested in the light itself and how it influenced plant growth. He began experimenting with different colored lights and their effects on plant respiration. the process by which plants convert sugars into energy. Blackman's experiments were conducted with a carefully designed apparatus involving a sealed glass jar, a small aquatic plant, typically Alodia, a common waterweed, and a sensitive instrument for measuring gas production. He noticed that when a plant was exposed to sunlight, it produced bubbles, oxygen, at a significantly faster rate than when exposed to darkness. Crucially, he didn't just observe this increase in oxygen production, he attempted to quantify it. Blackman developed a way to measure the amount of gas produced over time. He realized that the rate of gas production was directly proportional to the amount of light received. This was a crucial step, demonstrating a causal link between light and plant respiration. However, like Priestley before him, Blackman lacked a comprehensive understanding of why this was happening. He believed that plants were actively consuming carbon dioxide from the air, driven by the light, and releasing oxygen. This was a reasonable hypothesis given the limited scientific knowledge of the day. Blackman's meticulous measurements and systematic approach provided a stronger, more quantitative basis for Priestley's earlier observations. He moved beyond simply noticing changes in air quality to actively investigating the conditions that influenced those changes. His work, published in the early 1800s, contributed to a growing body of evidence suggesting a fundamental connection between plants and the air around them. Blackman's experiments laid the groundwork for future scientists like Jan van Helmont, who conducted a famous, though somewhat flawed, experiment involving a barrel of earth and a young oak tree, meticulously tracking the tree's growth while controlling the amount of water supplied. Van Helmont's observations, though not fully capturing the essence of photosynthesis, further solidified the idea that plants relied on external factors for their growth and sustenance. These early explorers, Priestley and Blackman, built upon each other's work and provided the initial clues that would eventually lead to the profound understanding of photosynthesis we have today. A story that truly began with a curious man and a jar of plants. The sunlight, as we've come to understand, isn't just a stream of photons, individual packets of energy. It's far more complicated and far more beautiful than a simple wave or particle. Within the leaves of plants, a truly astonishing phenomenon is unfolding, one that challenges our most basic intuitions about how light interacts with matter. This is the realm of quantum coherence in chlorophyll, and it's where the magic of photosynthesis truly begins. Chlorophyll, the pigment that gives plants their vibrant green color, isn't just passively absorbing sunlight. It's actively participating in a dance of probabilities, a state of being where multiple energy possibilities exist simultaneously. This is fundamentally linked to the concept of superposition. Think of a coin spinning in the air. Before it lands, it's neither heads nor tails, but rather a combination of both possibilities. Similarly, a photon of light striking a chlorophyll molecule doesn't just absorb one specific energy level. Instead, it exists in a superposition of absorbing multiple energies at once. The molecule briefly embodies the potential to accept energy at various wavelengths, creating a blurry, probabilistic landscape of interaction. This isn't merely a theoretical curiosity. Experimental evidence has repeatedly demonstrated that chlorophyll molecules can maintain this superposition state for remarkably long periods, even hundreds of femtoseconds, which is an incredibly short time. A femtosecond is 1/4 of a second. During this brief window, the the molecule is effectively exploring all the possible ways it could interact with light, vastly increasing the chance of a highly efficient energy transfer. It's like having a multitude of tiny antennae, each poised to capture a photon, but only one actually chooses to respond. The key to this efficiency lies in quantum coherence, the ability of these superimposed states to remain correlated. This means that the probabilities associated with each energy level aren't randomly fluctuating. They are linked together in a delicately balanced state. This coherence isn't spontaneous. It's driven by the interaction of the incoming photon with the molecule's electrons. The photon's energy momentarily excites an electron within the chlorophyll, and this excitation creates a temporary, correlated state among the molecule's energy levels. Now, let's delve a little deeper into how this correlated state spreads. Within the complex architecture of a photosynthetic reaction center, a specialized area within the leaf where light energy is converted into chemical energy, chlorophyll molecules aren't isolated. They are arranged in intricate antenna systems, forming chains and networks. It's within these networks where entanglement begins to play a crucial role. Entanglement is one of the most perplexing concepts in quantum mechanics. In essence, it describes a situation where two or more particles become linked in such a way that they share the same fate, no matter how far apart they are. Measuring the properties of one entangled particle instantly influences the properties of the other, a connection that seems to defy the limitations of the speed of light. In the context of photosynthesis, researchers theorize that entangled chlorophyll molecules within the antenna system act as a kind of quantum relay race. When a photon strikes the initial chlorophyll molecule, the excitation, and therefore the entangled state, spreads through the network via correlated transitions between the energy levels of the molecules. Instead of a single photon being absorbed and immediately used to drive the chemical reactions, the energy is effectively broadcast across the entire antenna, greatly increasing the probability of it reaching the reaction center, where it can ultimately be used to produce energy. Imagine a perfectly synchronized chain of dancers, each moving in perfect harmony with the others, even though they are spread across a large stage. The energy transfer is analogous to this coordinated movement, with each chlorophyll molecule contributing to the overall energy flow. The entanglement ensures that the energy isn't lost as it travels, maintaining its coherence and maximizing the efficiency of the process. The precise mechanisms governing this entanglement are still being actively investigated. Scientists are using sophisticated spectroscopic techniques, methods for studying the interaction of light with matter, to probe the dynamics of these quantum states. These experiments are pushing the boundaries of our understanding and revealing the surprising ways in which nature harnesses the principles of quantum mechanics. It's important to note that this quantum coherence isn't always present. When light is dim, or when the environment is noisy, with vibrations or other disturbances, the delicate quantum states can be disrupted and the efficiency of photosynthesis decreases. The plant, in a remarkable way, seems to actively manage this sensitivity, adjusting its molecular arrangement to maintain coherence under varying light conditions. Furthermore, the energy isn't simply transferred. It's ultimately converted into a specific, ordered state within the reaction center, preparing the plant to use that energy for growth and survival. The the quantum dance culminates in a precise outcome, demonstrating a stunning interplay between the probabilistic world of quantum mechanics and the deterministic world of biochemical reactions. Ultimately, the quantum leap reveals a profound truth: that the most fundamental processes of life are not governed by simple rules, but by the complex and often counterintuitive principles of the quantum realm. It's a reminder that the universe is full of surprises, and that even the smallest components of nature can possess the most remarkable capabilities. Deep within the green heart of a leaf, tucked away inside a specialized compartment called the thylakoid, resides a remarkable machine. It's not a machine built of gears and wires, but one constructed from intricately folded proteins and vibrant pigments, the reaction center complex of photosystem II. This complex is the crucial point where the energy captured from sunlight is finally put to work, initiating the chain of events that will ultimately convert light into the chemical energy that fuels a plant's life. The core of the reaction center complex is a tiny manganese-containing cluster, a relatively small group of atoms arranged in a precise geometry. This manganese cluster, along with calcium and chloride ions, forms the heart of the complex and is responsible for accepting and donating electrons. Around this central cluster, we find chlorophyll molecules, the pigments that initially absorb sunlight. These chlorophyll molecules aren't just passively absorbing light, they're actively participating in a dynamic, almost frantic dance. When a photon of light strikes a chlorophyll molecule, the energy from that photon excites an electron, boosting it to a higher energy level. This energized electron isn't stable in this excited state for long. It immediately seeks to return to its ground state, and it's this transfer of energy that drives the entire process. Now, let's zoom in on what happens next. The excited electron doesn't simply fall back to its normal state. Instead, it's pulled into a specific location within the manganese cluster. This isn't a passive absorption, it's a forceful interaction. The manganese ions within the cluster have a very specific affinity for electrons, almost like tiny magnets drawn to a particularly energetic piece. The excited electron from the chlorophyll, a molecule, is captured by one of the manganese ions, initiating a cascade of events. This initial capture isn't a single, isolated event. It's coupled with a process called proton translocation, the movement of hydrogen ions, protons, across the thylakoid membrane. The manganese cluster effectively acts as a proton pump. As the excited electron interacts with the manganese ions, it causes the release of protons from a small water-filled area associated with the complex. These protons are actively pushed across the thylakoid membrane, building up a concentration gradient. This gradient, with a higher concentration of protons on one side of the membrane and a lower concentration on the other, represents a stored form of potential energy, much like water held behind a dam. This proton gradient is absolutely critical. It's the driving force behind what's known as the chemiosmotic process, a fundamental principle of energy conversion. Think of it as a microscopic battery being charged. The flow of protons back across the membrane, driven by this potential energy, is then harnessed to generate ATP, adenosine triphosphate. ATP is the primary energy currency of the cell, fueling countless biochemical reactions. Specialized proteins embedded in the membrane, called ATP synthase, act like tiny turbines, using the flow of protons to spin and generate ATP. The manganese cluster isn't just involved in pulling electrons, it's also a key player in splitting water molecules. This is where the oxygen we breathe comes from. When water H2O is exposed to light energy, it's split into hydrogen ions, electrons, and oxygen. The electrons released from water replenish those lost by chlorophyll, and the oxygen is released as a byproduct. This process, known as photolysis, is an essential part of the entire photosynthetic cycle. The electrons released from water don't simply vanish. They are channeled through an electron transport chain, a series of protein complexes embedded in the thylakoid membrane. These complexes facilitate the transfer of electrons from water to the manganese cluster and ultimately to NADP+, an electron carrier molecule. This electron transport chain further contributes to the proton gradient, amplifying the flow of protons across the membrane and, consequently, the production of more ATP. The manganese cluster isn't a static entity. It's undergoing a constant series of redox reactions, changes in the oxidation states of the manganese ions. One manganese ion might be actively accepting an electron, while another is donating it. This dynamic exchange of electrons and protons is what keeps the whole process running smoothly, maintaining the proton gradient and driving ATP synthesis. The efficiency of the reaction center complex, and indeed the entire photosynthetic process, is truly remarkable. It's a delicate dance of electrons and protons, orchestrated by precisely arranged molecules and exquisitely controlled gradients. The manganese cluster, with its ability to both accept and donate electrons, is the core of this dance, tirelessly converting light energy into the chemical energy that sustains life on Earth. It's a miniature, incredibly sophisticated machine, operating within the quiet, green heart of a leaf, silently and powerfully fueling the world around us. The sunlight, having been brilliantly captured and organized by the quantum leaf, its energy now residing within the specialized structures we've explored, began its work in transforming something far more fundamental: carbon dioxide from the air. This wasn't a simple absorption. It was the beginning of a remarkably intricate biochemical process known as carbon fixation, and the star of the show was an enzyme called rubisco. Rubisco, short for ribulose-1, 5-bisphosphate carboxylase/oxygenase, is the most abundant protein on Earth, a testament to its crucial role. It's a massive molecule, a complex assembly of over 150,000 amino acids, and its primary job is to catalyze the first major step in converting inorganic carbon into organic molecules, and essentially pulling carbon from the atmosphere. The process begins with a molecule called RuBP, or ribulose-1, 5-bisphosphate. RuBP is a 5-carbon compound, and rubisco grabs a molecule of carbon dioxide, adding it to RuBP. This initial reaction, known as the carboxylate reaction, creates an unstable 6-carbon intermediate. This intermediate immediately breaks down into 2 molecules of 3-phosphoglycerate, 3-PGA, a 3-carbon compound. This is the first fixation. The carbon dioxide is now incorporated into an organic molecule. It's important to note that rubisco isn't perfectly specific. It can also react with oxygen. This is a significant problem, as oxygen reacts with RuBP in a process called photorespiration, which wastes energy and reduces the efficiency of the Calvin cycle. The 3 PGA molecules generated in this first phase then enter the core of the Calvin cycle, a cyclical series of biochemical reactions. These reactions occur within the stroma, the fluid-filled space surrounding the thylakoids inside the chloroplast. The first stage of the cycle involves phosphorylation, adding a phosphate group to 3-PGA. This is catalyzed by an enzyme called phosphoglycerate kinase. The resulting molecule is 1,3-bisphosphoglycerate. This molecule then undergoes a series of transformations, ultimately leading to the formation of glyceraldehyde 3-phosphate, G3P. G3P is another 3-carbon molecule, and it's the direct product of the Calvin cycle. Now, here's where things get even more sophisticated. For every 3 molecules of carbon dioxide fixed, 6 molecules of G3P are produced. This is essential because the Calvin cycle needs to recycle some of the G3P to regenerate RuBP, the molecule needed to continue fixing carbon dioxide. This regeneration process is incredibly complex, involving a series of enzymatic reactions. A significant portion of the G3P molecules, 2 out of every 6, are diverted out of the Calvin cycle to be used by the plant to build sugars like glucose and fructose. These sugars provide the plant with the energy and building blocks it needs to grow and thrive. The remaining 4 G3P molecules are then channeled into a complex series of reactions that ultimately lead back to RuBP. This regeneration pathway relies heavily on the enzyme ribulose 5-phosphate kinase. It's a key player, meticulously rearranging the molecules to restore the original RuBP. It's a highly regulated process, carefully controlled by the plant's needs, adjusting the rate of carbon fixation depending on factors like light intensity, temperature, and water availability. The plant can essentially fine-tune its carbon-fixing machinery. The entire Calvin cycle is driven by the energy captured in the light reactions. Specifically, the ATP and NADPH produced when water is split and sunlight is absorbed. ATP provides the energy, and NADPH provides the reducing power, the electrons necessary to convert the carbon compounds. These products of the light reactions fuel the steps within the Calvin cycle, effectively linking the captured sunlight to the creation of sugars. Furthermore, the process isn't simply a linear series of reactions. Feedback mechanisms are constantly at play, adjusting the efficiency of the cycle based on the plant's internal state. If, for example, the plant has a large supply of sugars already, the rate of carbon fixation may slow down. This intricate control ensures that the plant's energy and resources are used efficiently. The beauty of the carbon fixation pathway lies in its robustness and adaptability. Despite the challenges, the potential for oxygen interference, the need to manage energy resources, the Calvin cycle has evolved over billions of years, allowing plants to thrive in a diverse range of environments. It's a truly remarkable example of nature's ingenuity, a molecular dance driven by sunlight, carefully orchestrated by Rubisco, and ultimately responsible for the foundation of most food chains on Earth. It's a testament to the power of biochemistry, transforming the invisible gas around us into the very substance of life. The leaf, a vibrant emerald against the sky, isn't just passively catching sunlight. It's a master of efficiency, a tiny, intricate engine designed to squeeze every last drop of usable energy from the sun's rays. The initial capture of light, as we've discussed, is a remarkable process, relying on quantum mechanics to funnel photons into the reaction centers with astonishing precision. But that initial influx is only the beginning. A vast amount of energy, far more than needed for the immediate task of converting water and carbon dioxide into sugars, is inevitably lost as heat. It's a constant balancing act, a delicate negotiation between maximizing light harvest and minimizing these unavoidable losses. Within the leaf's mesophyll cells, the space between the broad, flat palisade layers packed with chloroplasts, lies a remarkable system of protective measures. The key is understanding that the energy a photon delivers isn't always used to build sugar. A significant portion, perhaps as much as 80%, is dissipated as heat. This isn't a failure. It's a crucial component of the leaf strategy. This excess energy, released in short bursts, fuels the dissipation pathways. Specifically, the cytochrome B6F complex, located within the thylakoid membranes. The cytochrome B6F complex acts as a kind of molecular heat sink. It's a series of protein complexes that accept the excess energy from photosystem II, rather than allowing it to cause detrimental damage to the photosynthetic machinery. These complexes effectively channel this excess energy into the movement of protons, positively charged ions, across the thylakoid membrane. This movement creates an electrochemical gradient, a difference in proton concentration, which is then harnessed to power the enzyme ATP synthase. ATP synthase, you'll remember, is the engine that generates ATP, the primary energy currency of the cell. However, simply generating ATP isn't the only way the leaf manages its energy. The dissipation pathways aren't just about producing ATP. They're also about preventing the overexcitation of chlorophyll molecules. Too much light at once can lead to what's called photoinhibition, a damaging process where chlorophyll molecules are broken down, effectively shutting down the photosynthetic process. The dissipation pathways provide a controlled outlet for this excess energy, preventing this destructive overload. The specific pathway used for dissipation can change depending on the light intensity. When light is abundant, the leaf preferentially uses a pathway that leads to the formation of nitric oxide, a small molecule that's readily released as heat. This safe dissipation route protects the photosynthetic apparatus from damage. Conversely, when light is less intense, the leaf might favor pathways that generate short-lived excited states, quickly decaying back to their ground state and releasing energy as heat. These pathways operate with remarkable speed, almost instantaneously. But the leaf's efforts to reduce energy loss don't stop at the thylakoid membrane. The structure of the leaf itself plays a significant role. The palisade mesophyll, arranged tightly in columns, maximizes the surface area exposed to sunlight. This dense packing ensures that each chloroplast has optimal access to light, minimizing shading and maximizing the chances of photon capture. Furthermore, the leaf's cuticle, the waxy outer layer, reduces water loss, which is vital for maintaining the proper biochemical environment within the leaf. Water stress can impair photosynthetic efficiency, making the plant more vulnerable to damage from excess light. The cuticle isn't just a barrier. It's a protective layer contributing to the overall health and resilience of the plant. The arrangement of chlorophyll molecules within the chloroplasts also contributes to energy efficiency. Chlorophyll molecules aren't uniformly distributed. They cluster around the reaction centers, where light capture is most effective. This concentration minimizes the distance photons need to travel to reach the reaction centers, reducing energy losses due to scattering and absorption by other molecules. It's important to understand that photosynthetic efficiency isn't a static value. It fluctuates depending on environmental conditions. On a bright, sunny day, the leaf operates at a high level of efficiency, maximizing sugar production. However, during cloudy days or in the shade, the leaf adjusts its strategy, prioritizing energy conservation over maximizing output. This adaptability is crucial for the plant's survival. The leaf's ability to dynamically respond to changing light conditions is a testament to the incredible sophistication of this natural process. It's a delicate dance between capture, conversion, and dissipation, a constant striving for equilibrium. The leaf isn't simply a passive recipient of sunlight, it's an active regulator, a master of energy management, quietly and efficiently transforming the sun's radiant power into the building blocks of life. This intricate system, built on quantum principles and fueled by a remarkable understanding of environmental cues, represents one of the most elegant and powerful processes in the natural world. And as the sun sets, the leaf begins to slow its activity, preparing for the night, but always holding within its green depths the potential for a new day of sunlight and sugar creation. If you're still awake and enjoyed this exploration, please consider liking this video and subscribing to Ultimate Sleep Doctor. Together, we can soothe more wandering minds one peaceful night at a time. The leaf's secrets now gently settle, a quiet dance of energy and light. Let your thoughts drift, calm and unhurried like the sun through the forest canopy. Allow yourself to surrender to the coming darkness. Good night.